डिजनरेशन से, न्यूरॉन्स की डिजनरेशन से लेकर के नेफ्रॉन्स की डिजनरेशन से लेकर के मैकुलर डिजनरेशन से लेकर के माने पूरे बॉडी के डिजनरेट को रिजुविनेट किया जा सकता है। ये पहली बार पतंजलि ने करके दिखाया। वी कैन रिजुविनेट लिवर, किडनी, हार्ट, लंग, ब्रेन एंड द होल सिस्टम। ये बड़ी उपलब्धि है। और टारगेटेड मेडिसिन के साथ साथ इंटीग्रेटेड मेडिसिन, प्लांट बेस्ड मेडिसिन से किया जा सकता है। यह भी पतंजलि ने करके दिखाया। सो वी आर प्रोवाइडिंग हियर टारगेटेड मेडिसिन, इंटीग्रेटेड मेडिसिन, और वी कैन रिजुविनेट द डिफरेंट ऑर्गन्स। और इसीलिए जब कोई पहली बार कोई चीज होती है तो उसके ऊपर आरोप प्रत्यारोप बहुत लगते हैं, हम उसकी परवाह भी नहीं करते। लेकिन नई दिशा ऐसे ही निकलती है। और लास्ट एक बात कह करके, इस धरती पर पूज्य आचार्य बागसिंह जी का और स्वामी रामदेव का जन्म और पतंजलि का ये संगठन साधारण काम को करने के लिए नहीं है, असाधारण कार्यों के लिए। इन हेल्थ, इन एजुकेशन, in research, in agriculture, in industry, everywhere we are doing the incredible work। और पूरी दुनिया पतंजलि को फॉलो कर रही है अलग अलग क्षेत्रों में। ऐसे इस संस्थान में आज विश्व के यहाँ पर गणमान्य साइंटिस्ट हैं। हमारा तो काम है सबको आशीर्वाद देना, उत्साह देना। अलग अलग यहाँ प्रेजेंटेशन होंगे। और अभी तक तो हम बगल में झोला डालते थे, आज अनुराग वार्ष्णेय जी ने हमारे गले में झोला डाल दिया। थैंक यू बहुत। मैंने कहा ये इतना लंबा चौड़ा पहचान पत्र तो पहली बार देखा। बोलो इसमें बहुत माल पानी भी रखा जा सकता है। तो ये अच्छा, नए नए क्रिएशन भी करते रहते हैं। हमारा डॉक्टर अनुराग वार्ष्णेय बहुत फनी भी है, वो लाफिंग और स्माइलिंग साइंटिस्ट है। अब जलेबी वाला कहना बंद कर दिया। बहुत लोगों ने इनके लिए जब आए तो जलेबी लाना शुरू कर दिया। तो मैंने कहा भाई ऐसा नहीं है, हमारा साइंटिस्ट अब पतला हो गया है। तो हम एक बार सभी साइंटिस्टों का और पूज्य आचार्य श्री जी के संकल्प का और सारे साइंटिस्टों के पुरुषार्थ का 7000 से ज़्यादा रिसर्च प्रोटोकॉल और लगभग 500 रिसर्च पेपर और अभी 500 की और तैयारी हो रही है। और ये सब कार्य खाली हमारे साइंटिस्ट ही नहीं कर रहे, यहाँ के BMS, BNYS और PhD और अलग अलग प्रकार के जो है शोध अनुसंधान के छात्र, वो भी इन रिसर्च प्रोग्राम्स में सम्मिलित हो रहे हैं। आप सबका भी एक बार बहुत बहुत अभिनंदन है। तो आप सबको ये जो रिसर्च का एक्सपोजर है, बहुत से इंस्टीट्यूट्स में तो पूरी पढ़ाई होली साहब, BMS कर लिया, BNYS कर लिया, PhD भी कर लिया, कोई डीलिट भी कर लिया, अलग अलग प्रकार के और बहुत सारे काम कर लिए। पतंजलि में जो बालक 1 साल रह लेता है, वो धरती पर जितने भी रोग और रोगी उन सबको देख ही नहीं लेता, उनका उपचार कैसे किया जाता है ये एक्सपीरियंस कर लेता है। और आप अपने जीवन में थोड़ा प्रयत्न करो तो कम से कम 100 रिसर्च पेपर लिख सकते हो। इतनी कैपेबिलिटी आपको पतंजलि में रह करके आ जाती है। ये डॉक्टर अनुराग कई बार कहता है कि कोई साइंटिस्ट जीवन में एक मेडिसिन को रिसर्च कर ले या इनोवेशन कर ले तो उसको लगता है मैंने जिंदगी में बहुत बड़ा काम कर दिया। यहाँ तो 100 से ज़्यादा बड़ी बीमारियों के लिए ऐसी औषधियाँ जो कि मॉडर्न मेडिसिन में भी जो पॉसिबल नहीं था, वो इम्पॉसिबल कार्य यहाँ पर पॉसिबल हुआ है। इससे बड़ी और उपलब्धि हो क्या सकती है। तो बहुत बहुत धन्यवाद सभी को और पूज्य आचार्य श्री को और पूरी टीम को प्रणाम।
परम पूज्य महाराज जी, आपके प्रेरणादायक मार्गदर्शन के लिए हम आपका हृदय से धन्यवाद और सादर नमन करते हैं। अब मैं आयुर्वेद शिरोमणि परम श्रद्धेय आचार्य बालकृष्ण जी से सादर निवेदन करती हूँ कि वे मंच पर स्वागत उद्बोधन से हम सभी का मार्गदर्शन प्रदान करें।
चलो जी, आचार्य जी के लिए बहुत बहुत अभिनंदन।
भारत माता की वंदे। आज पतंजलि का यह पवित्र परिसर परम श्रद्धेय पूज्य स्वामी जी महाराज की पावन उपस्थिति, उनकी प्रेरणा और हमारे इस मंच पर और मेरे सम्मुख भी विश्व के अनेक देशों से पधारे हुए हमारे वैज्ञानिक विद्वज्जन आदरणीय डॉक्टर प्रोफेसर डोमेनिको विटोरियो डेल्फिनो जी, आप पेरूजिया विश्वविद्यालय, पेरूजिया यूनिवर्सिटी इटली से यहाँ पर पधारे हैं। ये प्रेसिडेंट हैं इंटरनेशनल सोसाइटी फॉर इथनोफार्माकोलॉजी ISE के, और इथनोफार्माकोलॉजी के ऊपर आपको दुनिया जानती है और पतंजलि के प्रति और श्रद्धेय स्वामी जी के कार्यों के प्रति आपकी बहुत ही एक प्रेम और सौहार्द है। तो आपका बहुत बहुत अभिनंदन। प्रोफेसर डोमेनिको जी को बहुत ज़ोरदार एक बार आप करतल ध्वनि से स्वागत करें। और उनके साथ में बैठा हुआ ये जवान सा छोरा हमारा, कल हमारी बड़ी दोस्ती हो गई इसके साथ में, और ये डॉक्टर प्रोफेसर लुई फेलिक्स नाटियस, ये इनकी माता जी भी बैठी हैं इधर। ये माता बहुत अच्छी फोटोग्राफर है, तो मुझे दो तरह से अच्छा लगा। एक तो अपनी माँ को लेकर के घूम रहे भाई साहब, और दूसरा ये माँ बहुत ही अच्छी हैं। तो ये कोट द अजूर यूनिवर्सिटी फ्रांस से ये हैं, तो इनको बहुत ही हमारी शुभकामनाएं हैं। ये AI पर भी काम करते हैं और अलग अलग क्षेत्रों में ये काम करते हैं। और इसके साथ में यहाँ पर हैं आदरणीय डॉक्टर तिडारेट टुआंगयोट, ये बहन जो है हमारी डुआंगयोट जी, ये माइफा लुआंग यूनिवर्सिटी थाईलैंड से हैं। तो इनको बहुत बहुत हमारी शुभकामनाएं हैं। ये लुई फेलिक्स जी भी बहुत सारा काम करती हैं, प्लांट्स को लेकर के, बाकी चीज़ों को लेकर के यहाँ पर पधारी हैं। तो हमारे साथ साथ में यहाँ पर प्रोफेसर रूडोल्फ बाउर जी यहाँ पर हैं, ग्राज यूनिवर्सिटी ऑस्ट्रेलिया से। तो ये भी इथनो फार्माकोलॉजी के बहुत रिनाउंड साइंटिस्ट और प्रोफेसर हैं। तो प्रोफेसर रूडोल्फ बाउर को भी हमारी बहुत बहुत शुभकामनाएं हैं। और उनकी मुस्कुराहट भी बड़ी प्यारी है, जितना मैं बोल रहा हूँ सब समझ में आ रही है उनको, ऐसा मत सोच लेना नहीं समझ रहे हैं। साथ में हैं प्रोफेसर एन मेरी शॉनी पोर्डान जी, तो इनको भी हमारी बहुत शुभकामनाएं। ये सीनियर पब्लिशर हैं फार्माकोलॉजी एल्सवियर नीदरलैंड से। जो एल्सवियर का जो फार्माकोलॉजी वाला जो न्यूज़ जर्नल छपता है, उनके ये चीफ एडिटर हैं जहाँ तक मुझे पता है। तो मेरी बहुत बहुत शुभकामनाएं हैं इनको। और हमारे इस सब कार्य के कर्ता धर्ता हमारे प्रोफेसर पुलक के मुखर्जी जी, तो ये बहुत जुड़े हुए हैं सोसाइटी फॉर इथनोफार्माकोलॉजी SFE, ये जादवपुर यूनिवर्सिटी कोलकाता से हैं। तो इनके ही प्रयास से यह आयोजन यहाँ पर हो रहा है। और हमारे सुपरिचित पतंजलि, जिनको बहुत अच्छी तरह से पहचानता है और वो पतंजलि की गतिविधियों को पहचानते हैं, प्रोफेसर YK गुप्ता जी, जो एम्स कल्याणी भारत में अभी आप कार्य करते हैं। और हमारे बीच में हैं प्रोफेसर हमारे डॉक्टर MD रे, ये भी एम्स नई दिल्ली से हैं। और प्रोफेसर साहब हमारे क्लिनिकल रिसर्च में बड़ा योगदान दे रहे हैं। और आपका बहुत बड़ा पुरुषार्थ है कि हम लोग अलग अलग तरह के रिसर्च के कार्यों में बहुत कुछ कर पा रहे हैं। और साथ में इस मंच पर हमारे डॉक्टर अनुराग जी और डॉक्टर वेद प्रिया जी, दोनों को भी हमारी शुभकामनाएं। बात यह है कि इथनो फार्माकोलॉजी को लेकर के इंडिया में एक बड़ा इंटरनेशनल कॉन्फ्रेंस हो रहा है नाईपर चंडीगढ़ में। कहानी शुरू होती है यहाँ से। तो इथनो फार्माकोलॉजी की जब बात हुई कि नाईपर में हो रहा है, और हमने डॉक्टर मुखर्जी जी को, क्योंकि हमसे पहले से ही जुड़े हुए हैं, हमने कहा कि आपके पास इंटरनेशनल बड़े बड़े साइंटिस्ट आ रहे हैं, रिसर्चर आ रहे हैं, प्रोफेसर्स आ रहे हैं, तो उनको वाया पतंजलि करके कार्यक्रम तय किया जाए। तो डॉक्टर पुलकित जी ने कहा कि ये तो बहुत अच्छा रहेगा, क्योंकि इथनो फार्माकोलॉजी के ऊपर जितना पतंजलि में प्रैक्टिकली ग्राउंड पर जो रिसर्च हो रहा है वर्ल्ड में कहीं नहीं, और वहाँ के वर्ल्ड के साइंटिस्ट को पतंजलि जरूर दिखना चाहिए, इसीलिए यह आयोजन संभव हो पाया। अन्यथा इतने सारे साइंटिस्टों को एक जगह लेकर के आना वर्षों की योजना होती है, और उसके लिए श्रम के साथ में अर्थ भी बहुत लगता है। तो मुखर्जी जी के प्रयास से हमें सहजता से प्राप्त हुआ। तो इथनो फार्माकोलॉजी बियॉन्ड टाइम्स के ऊपर हमने कहा हम बात करेंगे, क्योंकि पतंजलि अभी प्लांट्स की हिस्ट्री के ऊपर, मेडिसिन सिस्टम की हिस्ट्री के ऊपर काफ़ी कुछ काम कर रहा है। जब मानव सभ्यता की बात आती है, इतिहास की बात आती है, तो हम लोग कहीं ना कहीं अपने आप को 2-4-5 हज़ार वर्षों में सिमट करके रह जाते हैं। तो जब हम 2-4-5 हज़ार वर्ष में सिमटते हैं तो सारी पैथियाँ, पद्धतियाँ, सारी समझ, जो हमारी प्राचीन इतिहास, मान्यताएं उतने ही वर्षों में सिमट करके रह जाती हैं। परंतु पतंजलि जो काम कर रहा है वहाँ पर हम मनुष्य सभ्यता को लगभग 28 लाख वर्ष पीछे तक हम ले जा चुके हैं। इसीलिए हमने कहा कि जब बियॉन्ड टाइम्स की बात होगी, इथनो फार्माकोलॉजी की बात होगी, तो पतंजलि बहुत महत्वपूर्ण भूमिका निभाएगा। कल इसके लिए हम बड़ी चर्चा करेंगे, और डॉक्टर रश्मि जी बड़ा काम कर रही हैं। मैंने उनको कह दिया था पूरी तैयारी कर लें, तो कल आप सबको बहुत विशेष रूप से पतंजलि के माध्यम से हम क्या कर रहे हैं उसको सुनने और जानने को मिलेगा। मैं तो आज सोच रहा था, जब इथनो फार्माकोलॉजी की बात है, तो हम जब नॉर्मली जब आप पढ़ते हैं ना स्कूल कॉलेज में, कहा जाता है कि मनुष्य को, ह्यूमन इज़ सोशल एनिमल, तो मनुष्य जब सामाजिक जानवर हो तो जो सोच जो है ना वो देवत्व वाली नहीं हो सकती है। तो हमारी संस्कृति कहती है अहम ब्रह्मास्मि तत्त्वमसि, या हम कहते हैं कि हम ईश्वर के प्रतिनिधि हैं, ईश्वर के पुत्र हैं, या ईश्वर के रूप हैं। उस संस्कृति से ही उसके सही स्वरूप को जाना जा सकता है। तो यहाँ से जो जानने की बात है, समझने की बात है, हम उसको दुनिया में पहुँचाने का प्रयास करेंगे। दूसरा, हम कहते हैं कि मनुष्य तो बंदर की प्रजातियों से बढ़ कर के फैल कर के आया, तो जानवर हैं और बंदर हैं। तो हमारी सोच पुरानी, वो जब दिमाग के अंदर ऐसी चीज़ें बैठ जाती हैं कि पहले तो हम ऐसे मूर्ख रहे होंगे, फिर हम लोगों को बता दिया गया कि ये पाषाण युग था, ये लौह युग था, ये धातुओं का युग था। जब इस तरह की अवधारणाएं, शिक्षाएं, इतिहास हमको बताया जाता है, सिखाया जाता है, समझाया जाता है, पढ़ाया जाता है, कहीं ना कहीं हमारा मन, हमारा मस्तिष्क, हमारा दिमाग इस तरह के सोच और विचार और भावनाओं से ही चलने लगता है। तो उन भावनाओं से परे भी कोई भावना है क्या? उन सिद्धांतों से परे भी कोई सिद्धांत है क्या? क्या इनसे परे भी कोई मान्यता या कोई प्रमाण अवेलेबल हैं क्या? यदि उन प्रमाणों को यदि हम दुनिया के सामने रखते हैं तो दुनिया निश्चित रूप से उन प्रमाणों को लेकर के विचार करेगी, सोचेगी और उस क्षेत्र में काम करेगी। इस भाव से ये इथनो फार्माकोलॉजी बियॉन्ड टाइम्स की हमने परिकल्पना की है। यह एक बहुत बड़े सोच के साथ में, बहुत बड़ी दृष्टि के साथ में आने वाले भविष्य के लिए नए, कहीं ना कहीं हम नए सृजन और नए शोध और नए सोच को पैदा करने के उद्देश्य से इसकी परिकल्पना की गई है। तो यह आयोजना एक बहुत बड़े लक्ष्य को लेकर के, तो हमारे मन में था कि आप सब हमारे प्राध्यापक गण, हमारे स्टूडेंट्स विशेष कर करके जो मेडिकल प्रोफेशन में जो बीएमएस कर रहे हैं, बीएनवाईएस कर रहे हैं, उन्हीं बच्चों को हमने कहा कि आपको इसको जानना, सुनना और समझना ज़रूरी है। एक एक साइंटिस्ट को सुनने के लिए, समझने के लिए आपको बड़ा मुश्किल होता है। इन लोगों से आप सब मिल भी पाएँ तो यहाँ श्रद्धेय स्वामी जी की कृपा है, उनका आशीर्वाद है। यद्यपि थोड़ा सा टाइट हो रहा बैठने में, श्रद्धेय स्वामी जी ने कहा कि टाइट ज़्यादा भरे भरे से लगोगे, तो भरे भरे से लग रहे हैं महाराज जी। यहाँ देखो आप, ये इधर कुर्सियाँ पे डलवानी पड़ी, डलवा दी गई है, तो सब आनंद से बैठ करके इसका आनंद ले लो। और ये मुझे कहते हुए खुशी है कि लगभग मैं पहले ही बता देता हूँ कि 2 दिन के बाद में जितने भी यहाँ पर साइंटिस्ट हैं, अभी तो ठीक है हमारी बात को ये कुछ समझ रहे हैं हमारे वाले, बाकी कम समझेंगे, बाद में ये समझा देंगे। मैं इतना ज़रूर कह सकता हूँ कि जिस भाव को लेकर के यहाँ से आए थे, उस भाव को लेकर के यहाँ से नहीं जाएंगे, इनके भाव बदले हुए पाएंगे। ये सामर्थ्य है पतंजलि का, ये सामर्थ्य है श्रद्धेय स्वामी जी का, और उसके लिए सामर्थ्यशाली कार्य का। तो हमारी टीम ने, हमारे वैज्ञानिकों ने, हमारे साइंटिस्टों ने जो काम किया है वो अद्भुत है, वो विश्वस्तरीय है, विश्व के जनमानस को हमारी ओर खींच लाने में सामर्थ्य रखता है। तो सबको मेरी बहुत बहुत शुभकामनाएं, सबको बहुत बहुत बधाइयाँ।
आपके अमूल मार्गदर्शन और उत्साहपूर्ण आशीर्वचनों के लिए परम श्रद्धेय आचार्य जी का हम हृदय से आभार व्यक्त करते हैं तथा आपको सादर नमन करते हैं।
Now we would like to felicitate the companions accompanying with our honorable guests on the stage. First of all, I would like to call on the dais Mrs. Christina Boyle, Mrs. Anisha Ray, Mrs. Rita Scaglia, Mrs. Pulok, Mrs. Mukherjee, सुनो शुरू करना। अब हम एक छोटा डॉक्यूमेंट्री पेश करना चाह रहे हैं। चर्चक भी आ गया। I would also like to invite Professor Sanjay Jatak on the dais for felicitation by Maharaj Ji and Param Pujya Acharya Ji.
थैंक यू। योग और आयुर्वेद के माध्यम से मानव के दुखों को दूर करने के एक महान और दिव्य संकल्प के साथ परम पूज्य स्वामी रामदेव जी महाराज एवं परम श्रद्धेय आचार्य बालकृष्ण जी निरंतर पूर्ण समर्पण, तपस्या और अटूट परिश्रम के साथ कार्य करते रहे हैं। उनके दूरदर्शी नेतृत्व, प्रेरणादायक मार्गदर्शन और अतः परिश्रम के कारण आज योग आयुर्वेद को विश्व भर में करोड़ों लोग श्रद्धा और विश्वास के साथ अपना रहे हैं। इसी सोच के साथ वर्ष 2017 में पतंजलि रिसर्च फाउंडेशन की स्थापना की गई। इसका उद्देश्य प्राचीन ज्ञान और आधुनिक विज्ञान और तकनीकी से जोड़कर आयुर्वेद को पूरे विश्व तक पहुंचाना है। आइए इस आधुनिक अनुसंधान संस्थान और इसके उद्देश्य के बारे में एक छोटी डॉक्यूमेंट्री जर्नी ऑफ पतंजलि रिसर्च फाउंडेशन एंड इंट्रोडक्शन के माध्यम से जानते हैं।
Under the visionary leadership of Param Pujya Swami Ramdev Ji Maharaj and Param Shraddhe Acharya Balakrishna Ji, the Patanjali Research Foundation was established in the year 2017. The goal of this organization is to connect our ancient wisdom and knowledge with modern science and technology and convey this information globally. Now, let us learn more about this advanced research institute and its mission through a short documentary titled Journey of Patanjali Research Foundation and Introduction. I would request the guests to go down. ठीक है.
आरोग्यम परमम भाग्यम, स्वास्थ्यम सर्वार्थ साधनम। स्वास्थ्य ही सबसे बड़ा धन है। यही वह आधार है जिससे संपूर्ण जीवन का निर्माण होता है। इसके अभाव में मनुष्य के सभी प्रयत्न अधूरे रह जाते हैं। हजारों वर्ष पहले जब विश्व की अनेक सभ्यताएं अस्तित्व की लड़ाई लड़ रही थीं, तब एक देश था भारत, जो वसुधैव कुटुंबकम की भावना को लेकर आगे बढ़ रहा था, जहां स्वस्थ जीवन का अर्थ रोग मुक्त शरीर ही नहीं बल्कि जीवन का पूर्ण संतुलन था। यह जीवन को समझने, जीने और संतुलित रखने का विज्ञान था। सनातन चिकित्सा पद्धति आयुर्वेद मात्र एक चिकित्सा पद्धति नहीं थी। इसने मानव को सिखाया कि स्वास्थ्य शरीर, मन, इंद्रियों और आत्मा का सामंजस्य है। महर्षि चरक, सुश्रुत और वाग्भट आदि महान ऋषियों ने विभिन्न प्रयोग,
चलो इसको बंद करो।
और
so we will
अनुभव के
send for
आधार
you
पर
the English version of this documentary. So we are going अभी नीचे आगे का प्रोग्राम शुरू करते हैं। इसको इंग्लिश में करना चाहिए था। ये तो जानते हैं अपने वाले, तो जिनको सुनाना चाहिए उनके लिए है। तो जब उनको समझ में नहीं आ रहा तो फिर जो है उसको चलाने का कोई फायदा नहीं। थैंक यू।
बहुत बहुत धन्यवाद। अब आगे के प्रोग्राम को हम प्रस्तुत करते हुए I would request all the dignitaries to please go down the stage and have your respective seats. Thank you very much for being on the stage. इसके साथ ही हम एक छोटी सी प्रस्तुति प्रेजेंट करेंगे। That is from Mr. Chandra Mohan ji. It is on the theme of P2P4. उन्होंने ये स्वतः बनाया है। It's a self-made song from Mr. Chandra Mohan ji. And he will be singing this song now.
उस पर नहीं चला कौन सा वाला
हो है माइक आप लोग ये वाला माइक बंद कर दो। ये माइक बंद कर दो।
ऑन म्यूजिक ऑन।
उपचार शोध से प्रमाण परंपरांग आधुनिक विज्ञान युगों से प्रवाहित अमर ज्ञान Plants to patients मानव कल्याण Plants to patients मानव कल्याण जनजातीय अनुभव ऋषियों की दृष्टि औषधि सूत्रों में जीवन की सृष्टि धरती की गहराई जड़ी बूटी का सार वनस्पति में निहित उपचार अपार लोक ज्ञान अब सीमित ना रहे वैज्ञानिक मानदंडों से बढ़े प्रकृति से उपचार प्रयोग से विश्वास ज्ञान मानव का साक्षात आधार युगों से प्रवाहित ये अमर ज्ञान Plants to patients मानव कल्याण Plants to patients मानव कल्याण फाइटोकेमिस्ट्री पर सूक्ष्म विचार बायो एक्टिव तत्वों का विस्तार क्लिनिकल परीक्षण सुरक्षा मान एविडेंस बेस्ड बने उपचार विधान लैब से लेकर क्लिनिकल केयर ज्ञान बने मानव सेवा का आधार प्रकृति से उपचार शोध से प्रमाण परंपरा संग आधुनिक विज्ञान युगों से प्रवाहित ये अमर ज्ञान Plants to patients मानव कल्याण Plants to patients मानव कल्याण आयुर्वेद आधुनिक प्रणाली एकीकृत हो बने नभ कहानी विश्व मंच पर गूंजे संदेश स्वस्थ भविष्य का होवे प्रवेश नव चेतना का स्वर जागे युगों का विज्ञान फिर हो आगे जो था प्राचीन आज भी सत्य समय बदले पर ज्ञान अमर तत्व प्रकृति से जीवन सेवा का पथ वो शोध सशक्त तो मानव हो स्वस्थ Plants to patient यही अभियान शरीर तो बने स्वस्थ समाज। Hearts to patients, यही अभियान। मानव कल्याण, यही अभियान। ओम प्रणाम।
मैं चंद मोहन जी और उनकी टीम को इतने सुंदर गायन के लिए बहुत बहुत धन्यवाद करती हूँ और मेरा आपसे अनुरोध है कृतल ध्वनि से एक बार पुनः उनका अभिनंदन करें। अब मैं इस अंतर्राष्ट्रीय सम्मेलन के विशिष्ट अतिथि आदरणीय प्रोफेसर वाई.के. गुप्ता सर को मंच पर पधारकर सेटिंग द टोन एथनोफार्माकोलॉजी एंड पतंजलि रिसर्च फाउंडेशन विषय पर विचार प्रस्तुत करने के लिए आमंत्रित करती हूँ। प्रोफेसर वाई.के. गुप्ता सर अंतर्राष्ट्रीय ख्याति प्राप्त क्लिनिकल फार्माकोलॉजिस्ट, प्रख्यात शैक्षणिक नेतृकर्ता जिन्होंने 40 से अधिक वर्षों तक न्यूरोफार्माकोलॉजी, क्लिनिकल फार्माकोलॉजी तथा टॉक्सिकोलॉजी का नेतृत्व किया है। आप ऑल इंडिया इंस्टीट्यूट ऑफ मेडिकल साइंस एम्स न्यू दिल्ली, एम्स जम्मू, एम्स भोपाल, इंडियन इंस्टीट्यूट ऑफ टॉक्सिकोलॉजी लखनऊ में डिपार्टमेंट डीन एंड डायरेक्टर के रूप में कार्यरत हैं।
May I now respectfully request Professor Y.K. Gupta sir to please initiate his lecture. Thank you.
परम श्रद्धेय श्री रामदेव जी, परम पूजनीय बालकृष्ण जी और यहाँ पर आए हुए several professors, dignitaries, scientists from across the globe, faculty and students here and the media people here. लोग कहते हैं कि दुनिया में हिंदुस्तान के दो लोग बड़े फेमस हैं, एक रामदेव जी, एक बालकृष्ण जी। रामदेव जी इसलिए फेमस हैं कि इन्होंने अपनी मेहनत से दुनिया को दिखा दिया कि योग से हम बढ़िया से बढ़िया इलाज कर सकते हैं कठिन से कठिन बीमारी का, और सस्ते में सुंदर टिकाऊ। और हर दुनिया के हर देश में, हर प्रांत में, हर नागरिक को उनका नाम पता है और लोग सम्मान करते हैं। बालकृष्ण जी ने बता दिया कि दुनिया में, हिंदुस्तान में कोई भी जड़ी बूटी ऐसी नहीं है, देयर इज नो हर्ब which does not have a medicinal property. Only thing is you have to discover and use it properly and scientifically. When there are two people like this who are respected, जिनका सम्मान करते हैं, आज जब उन्होंने हमारा सम्मान किया, यहाँ पे बैठे लोगों का सम्मान किया, तो सोचिए हम कितने बड़े बन गए। I think we are Indian, we are proud of such personalities who have raised the standards, the bar of Indian science, Indian ethos, Indian culture, Indian tradition globally. That is what something we are proud of you both, और हमें इस पे हमेशा गर्व रहेगा। मैं हमारे इन लोगों ने पता नहीं कैसी मेहनत करके एक ऐसे आदमी को चुन लिया जिनका नाम है अनुराग वार्ष्णेय, जो पहले बड़े जलेबी के फेमस होते थे। ये जलेबी हम जब भी आते थे तो हम इसीलिए आते थे कि अनुराग हमको जलेबी खिलाएंगे, लेकिन उन्होंने कुछ दिनों से जलेबी पूछना बंद कर दिया। आदेश, स्वामी जी ने आदेश किया कि अब जलेबी मत खाओ, तुम भी मत खिलाओ और डॉक्टर साहब को भी जलेबी मत खिलाओ, लेकिन फिर भी हमको प्यार से खिला देते हैं। ये प्यार ही है जो जोड़ता है एक दूसरे को। दिस इज लव, अफेक्शन एंड फेथ which connects the different pathies, which connects allopathy, which connects Ayurveda, which connects yoga, which connects all other Indian pathies together. Because what is objective is to treat, to maintain the holistic health of the humanity.
चाहे किसी भी तरह से करें. And you might have seen here in the stage, I am wearing coat, pant and suit and tie. On the other hand, there is a person just wearing one dress of simple nature, and दूसरे हमारे सिर्फ सिंपल सादी कुर्ता पजामा, और किसी ने अपना कुर्ता पहन के उस पर जैकेट ओढ़ ली. The only thing is everybody is wearing different, representing different culture, representing different Veshbhusha, but everybody is towards the health. And if I say the theme of this today's title is Ethnopharmacology Beyond Times, I was just connecting what is beyond time. And if you just see the knowledge का जो पाई होता है, एक चक्र होता है, एक गोला होता है, उसमें 3 खाने में अगर हम डिवाइड करें, one is I know, and this I know is nothing but ego, कि हमको पता है, अरे यार तुमको कुछ नहीं पता, लेकिन वो भी हमारा 0.001% उस पूरे गोल का होता है जिसपे हम घमंड करते हैं. I know. हमारे समाज में, हिंदुस्तान में ये आई नो काफ़ी बड़ा था, 1000, 5000, ये 1 लाख साल पहले, जैसे अभी हमारे आचार्य जी ने कहा. लेकिन हमने उसको अपने दूसरे देशों के इन्फ्लुएंस से हमने उसको भुला दिया. हमने कहा ये आई नो तो था, अब आई डोंट नो हो गया. उसके बाद एक दूसरा पाई आता है आई डोंट नो, ये हमको नहीं पता, और इसकी हम खोज कर रहे हैं, इसकी विज्ञान हम कर रहे हैं, इसकी खोज कर रहे हैं कि क्या ये ड्रग एंटी डायबिटिक में काम करेगी, कैसे काम करेगी, क्या एंटी कैंसर में काम करेगी, कैसे काम करेगी, क्या ये हर्बल ड्रग जो 10,000 साल से इस्तेमाल कर रहे हैं, अब हम इसमें पुनर्विज्ञान लेके आ रहे हैं कि इसमें कैसे और कैसे काम करे, कैसे मॉलिक्यूलर मैकेनिज्म से कैसे करेगी. दैट इज आई डोंट नो एंड नाउ आई एम ट्राइंग टू नो. लेकिन उससे बाद जो 80% इस पाई का हिस्सा है वो कौन सा है? वो है आई डोंट नो व्हाट आई डोंट नो. हमको पता ही नहीं कि हमको क्या नहीं पता, और वही काम यहां पे आचार्य जी और स्वामी जी कर रहे हैं. हमको वो पता करना है कि जो हमको पता ही नहीं है, जब हमको पता ही नहीं है क्या नहीं पता, यही हमको पता करने की कोशिश करनी है. पहले अगर हम ये पता कर लें कि हमको क्या नहीं पता, फिर हम उसको देखें कि क्या पता करें. तो ये ज्ञान के चक्षु खोलना जिसे कहते हैं, अगर हमें आ गया, हमें राइट टाइप ऑफ क्वेश्चनिंग करना आ गया, तो हमारी साइंस आ गई. हम हर चीज में हम साइंस जो हम कहते हैं पूछते हैं क्यों हुआ. एक अगर आपने हिंदी में गाना सुना हो, ये क्या हुआ, पता है ना, कब हुआ, कैसे हुआ, वो साइंटिफिक गाना है. ये कैसे, हाउ, वाई एंड व्हाट इज द मैकेनिज्म, क्या ये काम कर रहा है कि नहीं कर रहा, यही साइंस है. और हम इसीलिए जब हम कहते हैं कि जब हमारे विज्ञान हैं, 2-3 तरह के विज्ञान काम करते हैं. अगर हम कोई हर्ब या कोई मेडिसिन किसी में ट्राई कर रहे हैं, तो हम पहले देखते हैं कि डज इट वर्क, क्या ये काम कर रही है. तो हमें एपिडेमियोलॉजी लेके आनी होती है. हम ये नहीं कहते कि हमने इस महाराजा को ट्रीट कर दिया, इस किंग को ट्रीट कर दिया, 5 लोगों को ट्रीट किया. नहीं, विज्ञान चाहता है कि कितने सारे लोगों को आपने ट्रीट किया, कितने लोग उसमें से ट्रीट नहीं हुए, कितने आधे ट्रीट हुए, और उसके लिए क्या क्या आपने इन्वेस्टिगेशन किए जो कि इंटरनेशनल पद्धति से उसको मान्य हो. दैट इज व्हाट यू रिक्वायर साइंस टुडे. एंड आई एम वेरी प्राउड कि इसमें एक टाइम था जब हम आज से 20-25 साल पहले स्वामी जी से और बालकृष्ण जी से मिले, लोग चैलेंज करते थे कि ये डायबिटीज ड्रग ठीक करती है. स्वामी जी ने कहा कि ठीक करती है, लोगों ने कहा नहीं करती. फिर इसका जवाब एपिडेमियोलॉजिकल दिया गया, क्लिनिकल रिसर्च से दिया गया, और उसके बाद अब हमने मोस्ट मॉडर्न तरह के, चाहे इन विट्रो फार्माकोलॉजी हो, चाहे इन वीवो फार्माकोलॉजी हो, चाहे वो HPLC हो, चाहे HPTLC हो, मोस्ट साइंटिफिक, मोस्ट मॉडर्न इक्विपमेंट जो यहाँ पे पतंजलि रिसर्च फाउंडेशन में, जो मैं भी बहुत बार देख के सराहना कर चुका हूँ, वहाँ से हमने प्रूव किया. अब किसी को ये कहने की आवश्यकता नहीं होगी, हिम्मत तो लोग कर देते हैं, दुस्साहस भी कर देते हैं, उसको हम नहीं रोक सकते. लेकिन आवश्यकता नहीं होगी कि कहें कि साहब इसको दिखाओ करके। और इसका एविडेंस है, अगर आप जाके यहाँ के देखें लाइब्रेरी में, यहाँ की जो पब्लिकेशन का रिकॉर्ड है, मोर देन 700 इंटरनेशनल पब्लिकेशन फ्रॉम Patanjali Research Foundation. I salute those scientists working here. I salute those technicians working here. And I understand there are more than 250 patents here. There are more than 200 monographs जो कि पिछली बार हम आए थे तो बालकृष्ण जी का एक जन्मदिन था, उस पर उन्होंने दिखाया 200 मोनोग्राफ। एक एक मोनोग्राफ लिखने में लोगों को ज़िंदगी निकल जाती है उसका लिटरेचर कलेक्ट करने में। और ये सामूहिक collective effort, which I can say, have now you can go and get any reference of any these of the products. That is what science is. And it is important when you look for the herbal drugs or ethnopharmacology, botanicals, everybody talks of standardization of the product. वो कहते हैं कि this product requires standardization, कि आज गर्मी में जो बना, सर्दी में जो बना, पत्ते जब छोटे थे तब तोड़ लिए, पत्ते जब बड़े थे तब तोड़े, और किसी ने कहा 7 पुट्ट लगाया, सहस्त्र पुट्ट लगाया, 100 पुट्ट लगाया, इनमें क्या फ़र्क है? Therefore, the standardization is the global requirement. And I am very happy and proud to say that now today we have 500, more than 500 plants products का fingerprint है. Now if you have a 500 plants का fingerprinting, that means you can identify whether there is a purity 90% or 99% or 99.9% or it is 0%. And that makes a difference between the product which you sell from the Patanjali products and the other products. And that is the claim what you have. And that is, we are proud of that. Science is always evolving. And that's what we say, जो आज से हमारे पास 100 साल पहले हमारे पास प्रोडक्ट्स थे, चाहे वो च्यवनप्राश हो, चाहे वो कोई अष्टरिष्ट हो, कोई रिष्ट हो, चाहे कुछ भी हो, but then the formulation change, the improvement is the constant. Improvement change is the constant. If there is no improvement, there is no constant, and there is no constant, there is nothing dynamism. And that's why I'm proud to say that I was told by Anurag, 80 new formulations have been developed. 80 new formulations, whether it is for diabetes, whether it is for cancer, whether it is for pancreatitis, and whether it is for arthritis. अभी शायद वो फॉर्मुलेशन भी आ जाएगा जिसमें आप गंजे के भी बाल 1 महीने में उग जाएंगे, लेकिन अभी हमने ट्राई नहीं किया, इसलिए हम कभी कहते हैं अभी हमारी मैडम ने कहा अभी थोड़े और कम हो जाएं फिर, बट लेकिन ये कहना आसान है कि हमने फॉर्मुलेशन बना लिया. The more important thing that you have to have a clinical validation. You have to have a clinical trial. And I am very proud to say that now there are so many scientists here. There are scientists, more than 300 scientists working here. 100 PhD scientists working here. And there are scientists, more than 50 scientists coming from industry, pharmaceutical industry. There are scientists who are collaborating with the different academic institution, AIIMS, and collaborating for clinical trial, developing new protocol. These protocols are acceptable by the homeopathy, allopathy, or different disciplines, jahan kahan pe bhi ho. And as per regulatory requirement. The objective criteria, the end point which you set up for these clinical trials are those which are acceptable globally, those which can be published with international journals. And therefore, it becomes an evidence which is acceptable by science, by public, by regulator, and ultimately by the consumer. Ultimately, हमारा जो acceptability होती है, वो consumer की acceptability होती है. Any product, whether of any pathy, will not remain for longer period if this is not effective to the consumer for long. वो महीने, 2 महीने, साल भर करके बंद कर देगा. But if it is effective, it will go across the boundary, crossing the nation, and that's why you see the product which are Ayurvedic I am not naming this institution's name, it is understood, is regarded, respected everywhere in the world. And that is what हमको उसको बढ़ाना है. हमें ये भी दिखाना है कि हम नई नई खोज करते हैं. And if you just go, I wish that those who have come from outside, they must go and see Patanjali Research Foundation, the clinical trial facility, the outpatient department, the systemic documentation and the systemic analysis, statistical analysis. उनके जिसे कहते हैं, हिंदी में बोलते हैं, those who understand Hindi, चक्षु खुल जाएंगे. They will be mesmerized by the richness of the equipment. They will mesmerized by the richness of the rigor of the science which is being taken here. And that is what we look for, scientific evidence in this. And I am very happy that when ethnopharmacology, our Pulak Mukherjee and his society and international people have thought that let us go to Patanjali Research Foundation or Patanjali University. And it will not be surprising if they say after end of this meeting कि अब तो हम हर 2 साल के बाद Patanjali में visit करना चाहेंगे. To see the progress, to see how it is working. Lastly, I would say, whenever sometimes you speak slightly longer, आपने देखा होगा ना भारत में लोकसभा में कई लोग कहते हैं कि मेरा माइक बंद कर दिया इसका मतलब है that means the time is over. But I am very happy that there are several organizations which are doing MOU with this university, and I am sure that when you ये लोकसभा के स्पीकर ने आदेश दिया कि भी आप आधा मिनट बोल सकते हैं. हमारे 2 स्पीकर उपाध्यक्ष बैठे हैं सामने, इनके आदेश इन्होंने फौरन दिया कि बोलने दो इनको आधा मिनट और, तो मैं बोल रहा हूँ. तो ये एक I think we all should feel pride that we have gathered here for international meeting, not only to discuss what is being done, what should be done, but also importantly to showcase to the whole globe that we are number 1 in Ayurveda, we are number 1 in the world in exploiting the natures for the benefit of the human being at the cost which is affordable. Affordable cost आप समझते हैं ना कैसे हो सकती है, जहाँ पे अमिताभ बच्चन और बड़े बड़े दिग्गज लोग करोड़ों रुपए लेते हैं एडवर्टाइजमेंट के लिए, यहाँ तो हमारे स्वामी जी खुद ही एडवर्टाइजमेंट कर देते हैं एक कपड़ा पहन के. और इतना इफेक्टिव एडवर्टाइजमेंट इसलिए नहीं होता है कि उनकी शक्ल ऐश्वर्या राय से अच्छी है या उनके कपड़े बहुत अच्छे हैं, इसलिए कि वो बोलते हैं उसमें साइंस होती है, उसमें जन साधारण को ठीक करने की अभिलाषा होती है. एंड दैट क्रिएट्स मास अपील, एंड दैट इज व्हाट मास अपील का रिस्पेक्ट रखना हमारे जैसे साइंटिस्ट का धर्म है. टू कीप दैट फुल स्टॉप, दैट टू कीप दैट रिस्पेक्ट ऑन आवर सिस्टम is the responsibility of scientists, clinicians, clinical pharmacologists like us. And therefore, we assure you, Swamiji and Balkrishnan ji, that we will leave no stone unturned to prove what is correct, to prove what is valid, to prove what is effective, and to prove that the product which is made in India will match the global standards. Thank you. Jai Hind, Jai Bharat.
Thank you very much, sir, for this excellent overview of the journey made by Patanjali Research Foundation and making us feel proud of the achievement made till date. I would like to thank you, sir, with a clap. Before proceeding further, we would like to humbly welcome our respected Pujya Sadhvi Dr. Devpriya ji to this symposium, Ethnopharmacology Beyond Times, and request everyone to loudly clap their hands welcoming her. With this, we would like to proceed to the next lecture, and I would like to invite Professor Mukherjee to kindly come on the dais for presenting the main objectives of this symposium. Professor Mukherjee, sir, is serving as a full professor in the Department of Pharmaceutical Technology at Jadavpur University, Kolkata. He is also a former director of the Institute of Bioresources and Sustainable Development under the Government of India. Professor Mukherjee has more than 29 years of experience in evidence-based ethnopharmacological research and validation of metabolomics knowledge. He has published over 280 research papers and holds several patents. He is among the 2% listed global scientists published by the Stanford University and is a fellow of Royal Society of Chemistry, United Kingdom. I may now request Professor Mukherjee, sir, to kindly proceed with the objectives of this symposium. Please clap your hand, welcoming him.
परम पूज्य स्वामी रामदेव जी, परम आदरणीय बालाकृष्ण जी, and my friends from abroad, and respected Professor Y.K. Gupta and other colleagues from India who have joined to this Plan to Patient Series 4, being organized by Patanjali Research Foundation and the University of Patanjali as a satellite symposium for this 13th International Congress of the Society for Ethnopharmacology. Friends, it is my great pleasure to be associated with Patanjali Research Foundation, specifically to know more, or rather enrich my own knowledge with the Asirvad, the blessings from Swamiji and Acharyaji. Thank you. So, जो प्रोफेसर Y.K. गुप्ता जी वो हमारा भी गुरुदेव है. So, he is my teacher. So, what he told, no, that is the main thing. So when we talk about Plan to Patients, India's journey from ancient root to modern civilization, that has emerged in a very big way. And that is what Acharya Balakrishna ji in his talk, in welcome address, highlighted what Patanjali has done. Tomorrow you will be hearing the history. But India's rich heritage of traditional medicines represents a unique intersection of culture, biodiversity, and healthcare wisdom. And this is to maintain an unbroken continuum of the nature-inspired practice, which is deeply enrooted to this empirical knowledge and for thousands of years. Yes, we talk about validation, evidence-based validation, and so many things. I was just talking with morning, no, we are having a lot of discussions with Dr. Rudy Bauer and my good friend Dr. Domenico. Yes, we need validation, and at the same time, we have thousands years of evidence already in hand. So you have to give value to both. Yes, we need to prove what thousands years we are doing, that is true or not true, but that doesn't mean that thousands year knowledge in any way wrong. If we cannot prove it, that is, we are really, we should be blamed for that. That हम लोग ये prove नहीं कर पाया वो thousands years का history जो है, हम लोग का culture जो है, healthcare knowledge जो है, वो उसमें scientific validity नहीं है. इस तरफ नहीं कुछ अभी तक तो कुछ नहीं मिला. And Patanjali Research Foundation, yes, here only we got this ancient wisdom translated with scientific validation, with technological integrations, and they are showing a global leadership for this evidence-based traditional medicine model. You have already heard from Dr. Y.K. Gupta that this research foundation, which was founded in 2017 in the spiritual capital of India, the Haridwar, with the esteemed leadership of respected Swamiji and Acharyaji, and inaugurated by our Honorable Prime Minister of our country, Sri Narendra Modi ji. The whole thought is कैसे ancient wisdom का साथ modern science को bridge बना जाए. And really the publications, the patent, the work and this healthcare, wellness, product development, everything prove that the things are moving to the direct vision on that to be the global research foundations of the world in traditional medicine. And we feel proud with this core objective of this foundation, which is again the objective of our whole discussions on ethnopharmacology beyond times with the satellite symposium, is that evidence-based validations of this traditional Indian medicine and specifically development of global standards for Ayurvedic formulations, conservations and sustainable utilizations of the medicinal plants, and creating waste to wealth models for the agricultural sustainability and many more to prove that this is a global centers for evidence-based validation of Indian traditional medicine specially. Yes, this here is the essence, you know, whole India, the Atmanirbhar Bharat, and the startup and so many things are coming up. I do not know how many startup has come up from Patanjali Research Foundation. It may be thousands or 100,000 more, because anybody, everybody जो इधर education लेता है वो they are having their own startup, they are having their own mindset. So we are very much grateful to Swamiji and Acharya ji for making India proud. We all feel proud that tradition का translation जो real sense में होता है ये पतंजलि में ही होता है. And in fact without translation it is not an option now, it is an obligation. And when we talk about this ethnopharmacology beyond time, this is one of the major things that कैसे वो ट्रेडिशन को ट्रांसलेट करा जाए एंड इनको साइंटिफिकली वैलिडेट करा जाए. सो दिस गोल इज नॉट टू डिफेंड ट्रेडिशनल मेडिसिन्स बट टू स्ट्रेंथन इट, एंड इट रिक्वायर द ट्रेडिशन सर्वाइव्स बाय द विजडम and also by evidence, and we are translating the demand based on the regulatory aspect sometime. And here itself the answer is with Patanjali Research Foundation, and that is why we from society and everybody involved, including Domenico, our International Society Vice President is here, Rudy and other friends, whoever is here, everybody, we thought that the satellite symposium, this itself will showcase that how the tradition to translation is happening. And Patanjali Research Foundation is a example, real example for that in India and throughout the world. Here, drug discovery development, safety, toxicology, pharmacokinetics, bioavailability, anti-infective research, anti-aging geriatric research, metabolic disorders, nanotechnology, environment health, in different area is being focused to establish that how the molecule work and how the molecules together work. So दोनों का reductionist approach, synergistic approach, everything together is being explored in this Patanjali Research Foundation, for which we really feel proud that how this translation is going on. Now, the whole thought of our this year Congress in Naipar and also in this satellite symposium, that is why we start from here, that all our friends or the scientists and the students who are studying here, that when we talk about the translational ethnopharmacology, a Patanjali Research Foundation ही है जिसमें real sense में translational ethnopharmacology का काम हो रहा है. And this traditional, this translational ethnopharmacology approach is on 4 pillars. One is the traditional knowledge documentation, ethnobotanical, ethnomedicinal evidences drawn from living tradition and the historical text. I hope all my friends from abroad and India has visited the research center and also the herbarium. Dr. Bedpriya Acharya and our friends, Dr. Anurag, they have been taken a very great initiative. And so the first pillar, you will get all answer of that if you visit that place where the documented ethnobotanical and ethnomedicinal evidences drawn from living tradition and historical text. It is already evidence and from publication, patent and anything and everything what is there. Second aspect is the mechanistic validation. AV in vitro, in vivo characterizations or the bioactive constituents and molecular targets. It has been again proven in many different ways from the our ancient Ayurvedic text by Dr. Anurag and his group in this Patanjali Research Foundations. Third pillar is the clinical relevance, what Dr. Y.K. Gupta ji was highlighting, that evidence of therapeutic efficacy, safety, pharmacokinetics in human population. Yes, there is a very good group in this research foundation, and also they have collaborations with many institutes, organizations in India and abroad. And you will be hearing some of those stories from Dr. Ray and from AIMS as well. And the fourth, the most important pillar is the healthcare integration. What Pujaniya Swamiji was highlighting, no, that integrative research, it happens in India first time in Patanjali, and we are so proud that integrative hospital has been opened last month. It was inaugurated, and it is working in a very big way. So integration of this ancient and modern healthcare, including this regulatory acceptance, clinical guidance, incorporation, patient-centered application, everything is possible with this traditional ethnopharmacology. So friends, we talk about systemic ethnobotanical documentation, cross-cultural patterns or disease indication mapping, indigenous knowledge protocol, everything is possible through that, and which is again getting much more emphasized to the AI renaissance, modernizing the traditional medicine. This is another highlights what we are going to put on this digitizations of this ancient knowledge, predicting the hubs and the mechanisms with network pharmacology AI model, accelerating the natural product discovery, where the deep learning cheminformatics models will be provided, and also the quality and safety intelligence, including the sustainable bioresource management with remote sensing, AI forecast over harvesting risk and all these things towards precision and traditional medicines concept. So at the end, we all want to highlight that plan to patients where was the tradition and what is the trend. So trend has been set up by Patanjali Research Foundations for the translational research and also what we think ethnopharmacology beyond time. And we are really very much grateful and thankful for the enormous contributions of the scientists, technologists involved in this Patanjali Research Foundation for bringing together this evidence-based Ayurveda with different techniques where Society for Ethnopharmacology and the International Society for Ethnopharmacology is very happy to join. As you know, this society was rather established with the mentorship of our most respected former president of our country, Dr. APJ Abdul Kalam, who was the mentor, and with the mission to build a scientific bridge that honors traditional wisdom and uphold the rigor of modern validation, established in 2012 with this main motto of globalizing local knowledge and localizing global technology, vocal local. And that is our main aspects on that. And in fact, this society has a long presence in India with 18 local chapter, over 2,000 plus members, 12 of very impact studies we have performed, where 50 plus global knowledge outreach program and 15,000 delegates who join. We are having many programs, including the traditional healers program, which is one of the major thing where we promote the Lok Sastha Parampara. We have the codified system, but the non-codified system, how to promote. And that is one of the major thing we took forward on that, to highlight on that. And a very special thing what happened in this year with the respected blessings of our Swamiji and Acharya ji. We have been able to establish this journal with the system ethnopharmacology and sustainable bioresource from Elsevier, and Dr. Anne-Marie Pardon, she is here, who has made all these things in a very different way to establish that it is a first of its kind international journal dedicated to the holistic, evidence-based and sustainable aligned research with the bridging ancient wisdom with modern science. It is a new home for integrative research. And this is the first journal ever established in India by Elsevier to a society. So we are very much grateful and thankful, and Anne-Marie's first visit is here in Haridwar. And we believe that this journal with the blessings of Swamiji and Acharya ji will be able to cope up with the advances in a new paradigm in ethnopharmacology that connects plants, formulations, people, culture, ecosystems, and policies within a single system framework where we highlights everything, including the evidence-based validations, the bioeconomy development, the startups, the ethno-entrepreneurships, and many more. And also it aligns with the United Nations Sustainable Goals. So friends, we look forward that ethnopharmacology beyond times, and with this 30th International Congress, which we will be witnessing from 26 to 28 at Mohali. And also we have the Dr. Domenico will be highlighting, we are having in 2026 the IAC Congress in Italy. He will be telling more. And in 27, next year, we have a plan to host with over 50 countries together with different organizations of the world to highlight that how these translational traditional therapies can explore, educate, and empower, and how India can showcase the new route for development of these traditional medicines with these validations, which strengthen global collaboration, influence policy frameworks, advance evidence-based traditional medicine. So with this, I once again thank you very much. And in fact, for all our friends from abroad taking so much of pain, I know many of them, they just reached Delhi yesterday at 2 p.m., 2 a.m. in the night, means today early morning, and they are here today. So we are very much grateful and thankful to all of you who have joined, and I'm sure that this bioresource, which are the nature's gift for everyone, through that ethnopharmacology and the evidence-based validations, we will be able to promote and those gifts for everyone for the healthcare, and also preserve those gifts for the betterment of the healthcare of the society at large. Namaskar, Jai Hind.
I would like everybody to clap your hands, thanking Professor Mukherjee for presenting the symposium objectives clearly and highlighting the tasks done by the Society of Ethnopharmacology.
It gives me great pleasure and honor to invite Dr. Anurag Varshney sir, Vice President, Patanjali Research Foundation, to the dais for presenting an introduction of Patanjali Research Foundation. Dr. Varshney sir has obtained his doctoral from NCPSTFR Bangalore and his postdoctoral experience from Yale University, USA. He has more than 15 years leadership experience in reputed pharmaceutical company like Ranbaxy and Daiichi Sankyo. I now respectfully request Dr. Varshney sir to share the activities and achievement of the Patanjali Research Foundation. Kindly welcome him by clapping with your hands.
Om. I would request all of you go in the same sync as the Om that I will try. I am not the best at it, but I will request if we can have a deep breath and have a chant out of the Om so that we can connect with the same cosmos that we are sitting here at Patanjali. For the folks who has come from abroad, it's an humble request. Try it out. It's very, very relaxing, and it really connects you with the theme that we're going to go through. So let's start it. Thank you very much. So with my humble regards and bow in the holy feet of Param Pujya Swamiji, Param Shraddheya Acharya Ji, I would like to welcome all of you to this fantastic 1.5 day symposium on ethnopharmacology beyond times. And I'm very happy to have the lot of guests come from abroad who have taken their time to come and visit Patanjali, and I would like to welcome them by extending the heart feelings from Patanjali Research Foundation. And we have with us Dr. Domenico Delfino, who's sitting in the audience and who has been here earlier as well, a couple of years back, and we have been connected for quite a while, and we have been conducting some good research together as well. So let's clap clap for Dr. Domenico, who has been a fantastic colleague, fantastic scientist, and has been the president of the International Society of Ethnopharmacology. And we also have Professor Louis Félix Notias, who's sitting in the audience right over here. He looks very young, but full of energy, and has been exploring Patanjali for last 1.5 days, and he has been liking it very much too. So welcome, sir, to have you here with your mother, who's a fantastic photographer as well. And then we also have Professor Theodrat, who has come this morning at 2 p.m. from Thailand, and I think she's somewhere in the audience over there too. So thank you very much indeed for being here and being a part of this meeting. And then we have a couple of big stalwart of ethnopharmacology across the world. Professor Rudolf Bohr, sitting in the audience over there with her better half. Let's clap for him too, to welcome here at Patanjali Research Foundation. And then Professor Annie Mary Porden, who comes with a fantastic experience of working with a large publication house, Elsevier. Let's clap for him. And then we are missing 2 of our colleagues who could not come because of the visa issue they actually had it. We had Professor Jang Honji coming from Hong Kong. His visa got stuck at the nth hour. And we had Professor Satish Chandrabachar from Bangladesh, and he could not actually travel because of the political issues between those international borders. And then we have our mentors, Professor Y.K. Gupta sir, and I'm very thankful to him that he started the whole meeting so beautifully. Whatever introduction I wanted to give about Patanjali to our overseas guest, I think he covered most of it. So I would really like to thank him and would like to clap it as hard as possible. I think the students on the back can take out their hands from the phone and clap it faster, right? So that is for sir. And then we have Professor Pulak Mukherjee, who just set up the tone that what we're going to see. And then we have the audience that's sitting here, all the professors, and we have Puj Sadhvi Depriyaji with us, Professor Sanjay Chachakji, who's going to host a much bigger meeting, hopefully sometime soon. And then we're going to go through Dr. Depriyaji, my colleague from PRF, and then professors from university, professors from Ayurvedic College, and all the students from BAMS and BNYS course. All of you are here, and I'm very thankful. On behalf of the institution, I would like to extend the warm welcome and take it forward. So let's clap it for yourself, and then I'm going to start giving a little bit of introduction. So what I'm going to do today, I'm standing on a very tall platform, thanks to Acharji, because he gave that bridge to us so that I can connect. And I also see our respected Pujya Swami R. Devji in the audience, and I'm hoping to see Swami Bajrang Ji as well, both the service heads of Patanjali Wellness and Yogram, who has been doing fantastic things. So Swamiji, welcome for you to in this meeting. So what I'm going to give today, the audience who has come from outside and who are here for the first time in Patanjali, and those of you who have been part of it, for you it may be a slightly revision kind of thing. Take it like that when you take a flight, जब आप हवाई जहाज में जा रहे होते हैं तो you get a lot of security and announcement by the air hostess. You put a seat belt, do this, that. So for some of you, this might be like that security announcement. It is repetitive, but it's important. So please go by it, and I'm going to explain to you that what we do, how we do, what's the plan we have, what we have achieved, and which way we are going to go. I do speak sometime faster, sometime I also stammer, so I request all of you to bear with me, and if I get too fast, raise your hand, I'm going to slow down a bit. And we do have Professor M.D. Ray in the audience. I welcome him as well. So we have also planned 3 things here. So we are conducting clinical trials at 3 big locations. One is the AIIMS Rishikesh, there is one in AIIMS Delhi, which is just completed, and one in Tata Memorial Center. Other than that, we are also running 16 clinical trials. So these trials are going on. So we have the PI for those trials here, Professor M.D. Ray, and his better half is here. We would have Dr. Puneet Dhamija tomorrow, and we will also have Professor Bikram Gota from Tata Memorial tomorrow, who will then talk about what is their experience has been and how the drugs, the formulation which have been developed by Patanjali, being doing good in the clinical trial run by them. So let's welcome all of those as well whenever they are here. So to introduce PRF, Patanjali Research Foundation, we are blessed, influenced, guided, and directed by Param Shraddha Swamiji, Param Pujya Acharya on the daily basis, and on that direction, the PRF have been started. This whole setup started in 2017 and inaugurated by our respected Prime Minister Modi ji, and these labs have been set up to bridge the gap between the modern practice and the ancient medicine knowledge Ayurveda. They are probably one of the only kind, literally state-of-the-art facility to conduct the research on Ayurveda and yoga. Top-of-the-line equipments, globally trained workforce energize these operations on a daily basis. So we have like close to 300 scientists, and most of the scientists, the biggest toys that we want to have, जो हमारे बड़े खिलौने हैं, those toys are the equipments. If we have a good technology and good equipment, we can do wonderful science. So we are very blessed, and we really feel very fortunate from that we get a lot of those toys to play with, thanks to the guidance and support from Param Pujya Acharya and Swamiji, that we can do those experiments, and all of those equipment, all of those thoughts take the science in the right direction. Now, like any other institution, Patanjali Research Foundation has a mission, and the mission is to improve health and quality of life through research and innovation. That have been the mission of the institute, and we have been working very strongly about it. And now to do this, there is a vision that we really follow. So we already have an unstructured Ayurvedic and herbal information combined with the rich history of medicinal plant. And if you put a funnel of those scientific methods and combining the modern practice, larger scope of delivery, as well as the multi-pronged way of dealing with it, and go something about what we would like to do, a scientifically validated, evidence-based medicine system, what is called Ayurveda, which is accepted beyond the national boundaries of India or Southeast Asia, where Ayurveda is accepted, respected, and practiced as a mainstream medicinal system. That is the goal we are heading for. That's the vision that we're going to take it for. So I will request all of you for that vision, for that mission. Let's clap it hard. I'm going to clap with you. Now, when we talk about the research, and this is our main meeting that we're going to talk about, the research strand that Patanjali has, the strand that are there, which are there at Patanjali Research Foundation, the accreditation that we work with. So whatever is required to conduct a research in India and beyond, whatever the qualification, the quantifications, the regulatory compliance are needed, those are existing at Patanjali. This is a slightly busy slide, but I would like to take your attention towards that our laboratories are anyway accredited. They are accredited with DSIR, which is a government institution, Department of Scientific and Industrial Research. More than 300 scientists work here, several PhDs and postdocs with global experiences. We have a full-fledged animal house as per the CPCA requirement, governed by Institutional Animal Ethics Committee. We have a herbarium at PRF, which is accredited by New York Botanical Garden, USA. Our laboratories are in the compliance with Biosafety Level 2, which are again governed by the Institutional Biosafety Committee, and which have a dedicated clean and service corridor for microbiology and experimental animal facility too. On the top of it, we do a lot of clinical trials. We have two large clinical facilities in Patanjali Wellness and Patanjali Yogagram. On the top, we do multiple trials at external sites. Some of them are happening in India. 3 of them are happening outside India as well. Most of them are driven by Institutional Ethics Committee for Human Clinical Trials. And all the trials we conduct have been registered with the Clinical Trial Registry of India, what is called CTRI. And then what the good freedom we have, what the good liberty we have, our next door we have 250-bed Ayurvedic hospital with NABH accreditations, and which is an approved Ayurvedic medical college. Students are sitting here. You guys can clap for yourself as well for all the folks coming from this fantastic Ayurvedic medical college with the PG approval in the 8 subjects, and 2,200 Ayurvedic doctors across India at all those Patanjali Chikitsa Kendra, which has a daily footfall of close to 15,000 patients in a day. Now, see these numbers, 15,000 patients in a day, and if you combine them together, it comes to close to a 1 crore, 35 lakhs. Like, we are talking about 13 million patient's data are there in Patanjali electronic medical record, which is a humongous information. And as we say, data is money, data is information, information is future. So when you talk about this much of information about the patients and their treatment and all kind of modalities which have been all combined here. So this, if you see this whole slide in total, you would see what we have, a diversified research framework. That means that whatever is required to conduct the research, ethnopharmacology, herbal chemistry, herbal biology, whatever name we would like to take it, it actually either met here or Patanjali Research Foundation or have been built by the technical competency that we have. So when we talk about this foundation, so we have different segments to it. So we have our herbal research division, which is led by my colleague, Dr. Beth Priyareji. Tomorrow she's going to talk more about it and to explain the type of work that we do. That's, again, a fantabulous things. We have got the largest collection of plant paintings. We have got the largest collection of the compilation of medicinal information from the plants, which comes in form of a World Herbal Encyclopedia. We're going to hear about that tomorrow. We have plant paintings, we have a yoga research, and we also work on history and ancient documents. As Puj Acharji mentioned in the morning, that we worked a lot on history, how medicinal system evolved, how the things have changed from last several thousand years before. So those things are also very, very interesting and research driven. And we have our fantastic colleague, Dr. Rashmi Mittalji. She's going to talk tomorrow along with Acharji, and we're going to see the ancient documents and the history, what really happens. And then we get good support from new product development, Patanjali Ayurved Limited. We have Dr. Ram Gupta ji sitting here from the same. So thank you, Ram, for being here. And then from Central Labs and also linked from Patanjali Hospital and Medical College. And this is what I'm going to talk today. Drug discovery and development. So we have this as a very significant component of Patanjali Research Foundation, and that's where most of the folks who do a wet lab research at PRF work there. And I'm going to then introduce to you what we do here, what is the aim, and what are the modality we really work with. So these are just a glimpse of our infrastructure, the toys that I was talking about. So the PRF has, the drug discovery development has this 5 different components: in vivo biology, in vitro biology, herbal chemistry, microbiology, and clinical research. And the few of those pics that you see are right on the screen. And what we do, so when we started this whole operation, we had a long discussion with Param Puj Acharya, Param Shraddheya Swamiji, that how we're going to do. In Ayurveda, in the classical text, there are close to 1,000 formulations which are defined in the ancient old text. And there are close to 1,000 plants that we really work with. So if we combine them together, that becomes close to 2,000 test articles. Basically, in terms of research, you define everything you want to test as a test article. So there is a 2 way of doing it, that you take one test article, one drug, and start to look for the what disease it might work. That becomes basically a drug-driven approach or a formulation-driven approach. The other would have been to find a disease and see which formulation can work over there, what we call disease-driven approach, and we started working on the same line. Param Puj Acharya advised us, guided us, that let's go in this direction and find out if you go by a bucket of different therapeutic areas. For example, you look at inflammation, metabolic diseases, cancer, neuroscience, and infectious disease, and then you look for the type of disease you can work with and see whatever models you can set up. And then you test what are the formulation that you have with you, whether it's a drug working for diabetes, working for neurons, or any kind of pathology that you want to associate with. And these are the number what you see are the validation that we have already done. These are the really, really large number. And if you look at the success rate of these experiments, they actually meet any of the laboratories anywhere in the world. So for this success, let's clap for the scientists who worked very hard on those. So we work on inflammation. These are the disease, diabetes, and other stuff come in the metabolic disorder, cancer biology, neuroscience, and infectious disease, where we work with the bacterial, fungal, as well as viral diseases. During the COVID time, we were the first one to show that herbal medicines has an capability to actually fight with the bigger monster, COVID-19, and the whole team has actually taken off from there. And in the leadership of Dr. Sabita Lochabji right now, we are doing a lot of work in this area. And then we have different kind of biology function too. Now, this is my favorite slide, and this slide personally drives me a lot, and all of my able team that works it actually get driven by that. So if you see, we know what is pharmacology. So when we talk about modern drug discovery, जो नए दवाइयों बनाने का तरीका है, जो मॉडर्न मेडिसिन में सिंथेटिक ड्रग्स में बनाए जाते हैं, जब फार्मा तरीके से बनता है, इफ़ यू सी दैट टू गेट अ ड्रग टू द मार्केट फ्रॉम अ सिंथेटिक रूट, यू हैव टू गो दिस वे. This is called conventional path, that you make a compound, you make a molecule which is made by medicinal chemist, you test in the animals or in any models, and then you test in the man, what we call a clinical trial. This path is a conventional path, which takes close to $10 billion, 10 billion, not million, $10 billion and 10 years. And still there comes with lot of unsuccessful results and come with lot of adverse effect, the side effect what we call it. And the chances of success are also very low. So how do you going to deal with the disease burden that our society has? The one way to it, to go reverse path. Go it in the reverse path. We then can get inspired by the traditional knowledge, which actually the routine laboratory to clinic reverts to clinic to the laboratory. Why is that? Because we know from our ancient science, from our ethno practices, that Ayurvedic drugs, herbal drugs work. We know when we are going through a pains, we take different medications. When we're having difficulty, infections and fever, we take tulsi, we take giloy, all of that work, and which have been there in the practice for thousands of the years. So we know that efficacy exists, which is the biggest lacuna, biggest bottleneck when we go for a conventional path. So that would mean if you know efficacy, then it's just a matter of validation. All you have to do, validate it, find out where is that coming from, and then set up the safety. When people talk about therapeutic index, that means the ratio between the dose you get the adverse effect and the dose where you get a pharmacological effect. That thing is in the favor of the synthetic, that thing in the favor of natural medicine, Ayurvedic drugs. So all we have to do is start from there, which is the most important starting point, and validate the efficacy. Then you go to a reverse path. Then all you have to do, we have a data from the man, you go to the mice or any animal model and go to the molecule and find what are the players which are controlling the biology, whether it's happening at the level of the compound or it's happening at the level of the receptors inside the cells, whether it's happening inside the cell, on the cell surface, how the signal is getting affected, intracellular versus extracellular, all getting linked. And that's where we are going to work with. And that's take to my next favorite slide, where we talk about how do we strike the balance. How do we strike this balance between much needed need of the society of drug discovery. So this is my favorite slide. As I said, this is the triangle where you can combine Ayurveda with modern science. That means all the scientific advances that has happened for last 200 years. If you talk about X-rays, you talk about ultrasound, you talk about MRIs, these are scientific advances. Allopathic system happen to use them slightly better than Ayurveda have used. But if you combine them together, along with the modern medicine, which actually focus on evidence-based clinical practice, and then you would be able to set up this golden triangle. Now, those of you who are still awake and listening to me, and if you focus on this slide, if you eyeball this triangle, if you see this, you will find this triangle looks impossible at this moment, because all of these three arms of the triangles are not matching. Those who have seen it, let's clap for them, because I have seen it, right? So if you see this triangle looks impossible, but that is achievable. And if you see deliberately, I have put the logo of Patanjali Research Foundation, my institution, at the heart of this triangle, because we strongly believe in the guidance of Param Pujya Swamiji, Param Shraddha Acharya Ji, that we are going to meet this, reach it, and achieve it. Of course, Swamiji. So, okay, Swamiji. So traditional wisdom, research and evidence-based medicine, and that's going to lead it to there, Swamiji. तो हम यहाँ से यहाँ पहुँचेंगे. तो ये जो पूरा पाथ है हमारा, वो यहाँ से ही पहुँचेगा. सो आयुर्वेदा में ये है, एविडेंस स्वामी यहाँ से करेंगे कनेक्ट और इस ट्रायंगल को पूरा करेंगे. सो आइडिया ये है कि जो गोल्डन ट्रायंगल अप्रोच है ड्रग डिस्कवरी की, वो आयुर्वेदिक साइंस से पूरी की जा सकती है. अब इस काम को करने के लिए what we do here at Patanjali Research Foundation, the whole landscape of the work that we do, we go from plants to product and from product to the patients, which is the theme of this meeting. That how do we start from the plant, how do we go towards the product, and what are the steps that we take, what are the ways that we do. And in next 3 to 4 slides, I'm going to go very quickly through this, the technologies we use, the paradigm that we follow, and the protocols that we have. What is the SOPs for doing research going from plant to the product? So we start with the herbal chemistry, where we find out which plant to work with, plant selection, authentication, plant extractions, and biocompound isolation. Then we go to analytical chemistry and try to find out what are the signature fingerprints and find out what are the things that can be done. This is being led by Dr. Pradeep Nanji, who is the head of this function, and he has been in the audience and trying to set up this conference as well. Then we go towards biology. We do in vitro biology work, where you work on the 2D cells, 3D cultures, do a lot of cell-based assay, mechanism of actions, and molecular targets being seen, which becomes very critical to find out how drug works. That is being done, led by Dr. Rishabh Devji, and supported by Milsi ma'am and also Sandeep ji, which are there too. And then we go to in vivo biology, which is the fancy stuff, which is done by Dr. Sandeep Sinha ji and his team, then where we work on the efficacy, predominantly to find out if a drug works in what model, how that happens. That is being done. And then we move towards formulation. If things are good, then we complete the whole cycle, generate a formulation, and get it ready. And that's where the team from Dr. Nain comes back here, and we find out what are the requirements, what are the regulatory landscaping, and what can be done. In parallel, we also start the clinical research to find the mode of actions for the drugs, also the clinical relevance, which is being led by the clinical research team, and Dr. Momita ji lead that function. So I'm quickly go through each of those components. This is what we do for herbal chemistry: select the plants, find the solvent, get it done. Sometimes we do a supercritical fluid extraction, where we use liquidified CO2 to extract the best part of it, take the oily formulation, find out what is there, and then go for marker identification. And then we also go towards analytical chemistry, where we do HPTLC, high performance thin layer chromatography. All of these toys and tools are there in our laboratories. Some of you who are interested could visit and see those too. From HPTLC to UHPLC, LC-MS QTOF to UHPLC QTOF, MS-MS FID, which is gas chromatography, to MS-MS, to find out what is there in the air, which is a form of the volatile component, and what's in the liquid, which can be seen here too. And from there, then we go towards in vitro biology. That's where slightly more exciting things start to happen. You start to see the interactions of the herbal compound with the biological things. So you have cells that you do 2D cell cultures. You do a 3D cell culture when you make spheroids on them, which are slightly closer to the intact organs. And then we also do ex vivo models. That means the cells isolated from the patients, which are also grown. And then we do a very detailed biochemical molecular characterization, gene expression, changes at the level of the protein, changes at the level of protein phosphorylation, and there are other kind of changes that happen in the cells. And then we find out what's the mechanism of action. That means if we have in formulation and extract, what it works and how that works. I learned a very nice word from Puj Swamiji, Karyakari Siddhant, which is mode of action. So Karyakari Siddhant, we start to see from here that how these things work and in what principle things start to move in the right direction. And then get slightly more dynamic slides here, where you get to see the different model organisms that we work with. So we work with the C. elegans, which is a very elegant worms, which is a distant cousin of earthworms, and we have a dedicated laboratory where we have close to 116 types of C. elegans with us, which has genetical modifications, and you can use them in a very good model system for the disease. We do that. Recently, we have set up a Drosophila lab as well, which is called Drosophila melanogaster. Easy language called fruit fly is a fly that comes to your banana when you leave it on the kitchen, and that is a very good system too. And zebra fish, which are small, tiny fish. We are now making a new facility where fish and fly will stay together, and that facility would be also ready very soon. And in animal house, we have mice, rats, hamsters, guinea pigs, and rabbits, and we work with them to actually set up different disease model. These are all being done at Patanjali Research Foundation in Haridwar in these labs. We also work on dogs that we do a third party in Hyderabad, and we are also started to work with the monkey models that we do in China. So basically, whatever the requirement for a drug discovery is being done, met or bait here at PRF. And then we go towards the formulation. Once we find something, then we do a product optimization, find out what's the best way to deliver it. Is it a capsule, or is it a tablet, or is it a spray, or it's a topical ointment? Depending on the disease need and the patient compliance, we set up those systems, go towards the regulatory compliance and find out what are the needed to have it towards the market, what are the documentation that must be done, test the stability, scale it up, and transfer to our fancy team sitting at Padartha for the commercialization. And then comes to the clinical research. So that becomes one more critical component where we test the efficacy and safety, monitoring the patients, and also measure the outcome assessment. Not only for the disease, we also measure for the quality of life. We also measure the disease-related health quality of life and see what kind of changes happening in the life of the patients when they're going through a disease, and when they have been given these medications, Ayurvedic formulation, how those changes are being done, that get messaged too. And one thing I'm very proud to say, and which happened very recently, the Patanjali Research Foundation has been now accredited as a certified clinical research organization, a CRO, which is the first in the Ayurvedic industry. This is the first in the herbal space, and we are very proud that the whole team that has worked for it to set up that PRF is now a CRO, and a lot of things can be done under this umbrella and this accreditation. When we do all of these work, what are the outcomes we get? So the two-way possible. First, we develop a product which goes towards the market, which help the patients, which make sure that our society remain disease-free. The burden of the disease decreases. And what you see over here are those formulations. Close to 80 new formulations, 80 new medicines has been developed on the basis of research generated at PRF. So from head to toe, different organs, 5 vital organs, and 6 accessory organs if you see, from heart, brain, kidney, liver, lungs, everywhere, whatever the disease etiologies, pathologies possible can be addressed, treated, and cured by these formulations. Some of them have been really fantastic. Some of them has been totally novel. Some of them has been for the first time in the history of Ayurveda as well. For example, we have the formulation for pancreas, we have a formulation for prostate, which have never been thought of. And then Param Pujya Acharya Param Shraddha Swamiji has thought in those lines, developed those formulations, guided us in the right way of mixing those components, and telling us that this would be the most optimal way of getting it. And I'm very happy to see that 95 times out of 100 times, that's a 95% success rate, most of these formulations worked. So Swamiji, Acharya, thank you very much indeed for giving us that guidance, giving us that enlightenment to finding out different formulations. So this is what you see, which is from different body organs. And if you talk about the different physiological state of disease, whether it's actually pain, from infertility towards immunity to fevers and other things, and not to forget the cancer, but which I'm hoping that Dr. M.D. Ray will talk more about it, that all of these formulation works in those areas too. So the team has worked hard to reach to that level that we can validate the every stage of the research, can find out what drugs work at what level, at what disease pathology, etiology can be controlled and treated by these formulation too. So these are the one of the outcome we get from the research. The second outcome come from the high-end research paper. The drug discovery team alone has published in last 6 years 130 research papers with an impact factor. The total combined impact factor are close to 600. The average impact factor 4.5, which is really humongous. And I feel very proud standing here saying that this could not have been done anywhere else. As Swamiji says in the morning, that people who work on a single product, on a single drug or a single paper, they consider themselves that one PhD is done. Here, we are talking about 130 PhDs, or that too of the world-class work. So thank you very much, team, for being there and doing that too. And that comes to my final slide and come back to where I started from, the mission that we actually follow, that the mission that we have, we want to offer the whole world, the science of medicine from the oldest medicinal knowledge base, Ayurveda. and the mantra that we follow every day, Sarve Bhavantu Sukhna, Sarve Santu Niramaya. Thank you very much, team, and I really welcome all of you again, and I hope next 1.5 days you are going to enjoy and learn. Students in the back are going to learn a lot from international speakers with the experts who are going to share most of the things which they have learned, and I'm sure when they go back to their home, they will remember us, and we will also remember them and take this thing together. And I hope it's a good starting for a much bigger collaboration at a global level where PRF would be a big part of it. So thank you very much indeed. Swami Ji Acharya Ji, आपके चरणों में रखना.
Thank you, sir, for presenting the scientific vision, objective, and achievement of Patanjali Research Foundation. Now I request everyone to kindly please put your hands together for him.
Moving towards the keynote lectures, may I now request Professor Domenico Vittorio Delfino to kindly come to the dais for presenting his keynote lecture titled Uncovering Context-Dependent Immunomodulation, How Ficus rubiginosa leaf constituents influence the Treg Th17 axis. Professor Delfino is a distinguished physician and pharmacologist with over 30 years of experience in teaching and research. He is serving as the professor at the University of Perugia, Italy, and holds several other key important leadership positions. Currently, he is serving as the president and member of the board of directors for the International Society of Ethnopharmacology. Professor Delfino has published more than 106 peer-reviewed research papers and serves as associate editor for several international journals. I would request Professor Delfino to kindly proceed with his lecture. Thank you, sir.
Thank you. Thank you very much for this kind of presentation. And just a moment that we, okay, yeah, presentation. Presentation. Okay, ladies and gentlemen, first of all, I would like to thank very much the organizers for inviting me. I am indebted with Anurag for this, with Swami Ji and Acharya Ji. Thank you very much. You know, this is my second time in Patanjali, and I am so happy to be here again. I'm really happy because in the audience there are a lot of young people, a lot of students. I would like to be, yeah, I would like to be there with them, but I guess I'm too old. So anyway, with Anurag, Patanjali, and my university, University of Perugia in Italy, we have an MOU now, and so we hope that some students from Patanjali University can go to visit Perugia, our university, and maybe, Anurag, maybe this year someone can be, our student can be here. Okay, so what I'm going to do today is to tell you a story about a family plant. The family is Moraceae family, and to tell to you all this story, I would like to bring you with me for two travels. One is from India. Just a moment, please. The presentation.
Yes, sir, it's work.
Okay, it works now. What I'm going to do is to bring all of you with me for two types of trips. One is from India to the Far East, to the Vietnam, and the other is from India to Italy, of course, where I am from. These are the two trips. So let's start from going to Vietnam to talk about this plant family, the Moraceae plant family. One species in the Moraceae family is this tree. The name is Artocarpus tonkinensis. Is a Vietnamese tree, is endemic of the north part of Vietnam, where they know this tree as an ornamental plant. For example, a plant that is present in the garden of Buddhist pagodas. But in the very north side of Vietnam, close to the border with China, in a mountainous area, this plant is known as a medicinal plant because they use the decoction of the dried leaves of this plant as a treatment for arthritis and backache. This was discovered by Dr. Pham Chuc Lam during the war of Vietnamese against France. And the people that use the decoction of this plant for treatment is an ethnic minority, around 800,000 people living in that mountainous area around Son La, Lai Chau, Sapa, Cao Bang, Lang Son. And we know 5 branches of this ethnic minority, the Hmong, and they are called based on the colors of their dress. For example, white, red, green, flower, you see in the middle, these very colorful dresses. They are dressed by the flower monks. And this decoction of Artocarpus tonkinensis is used by the black monk to treat arthritis and backache. So we made some experiment to see if we can validate this knowledge, as Anurag said before, in a reverse pharmacology, let's say, pathway. And we have a very nice model, mouse model for arthritis, which is the collagen-induced arthritis. Yeah, you see my mice. This is an Italian mouse we call mouse Gigio, and we love it very much. So I gave to him this treatment because I was certain that was a safe treatment. Anyway, when you treat the mice with collagen and then you challenge again with collagen after 28 years, these mice develop arthritis. And they develop arthritis because they produce Th17 cells that goes by the attraction due to the chemokine CCL20 to the joints, and also because macrophages become osteoclasts that destroy the joints bringing to arthritis. So Th17 cells and osteoclasts are very much central for the pathogenesis of arthritis. So we gave to these mice the decoction of Artocarpus tonkinensis, and as you can see here, their swollen joints, paw, were completely reversed by the treatment with the decoction. And also at the histology level, if you can see, in a healthy mouse, you have a very smooth cartilage and no inflammatory infiltration in the joints. But if the mouse develops arthritis, the cartilage is broken and you have a lot of inflammatory arthritis. If these mice drink the Artocarpus tonkinensis decoction, you can see that again the cartilage is smooth and you have only a small inflammatory infiltration. So this decoction is really useful, as demonstrated by these experiments. We also made a gene expression profile of the joints, and as you can see here, you have a complete shutdown of interleukin 17 and interleukin 22, a downregulation of interleukin 6, of CCL20, the chemokine, of CCR4, the receptor for that chemokine, and interleukin 23. All these molecules are characteristic of a type of cell, which is the Th17 cell, which we have seen before is important in the pathogenesis of arthritis. That the decoction was going to inhibit the development of Th17 cells. We could see also by analyzing the paws, the lymph nodes, and the spleen of that mice, as you can see here by real time, there is a significant downregulation of interleukin 17 and the interleukin 22, the two cytokines characteristic of Th17 cells. So we can conclude that the decoction of Artocarpus tonkinensis leaves improves signs, symptoms, and histology of arthritis, since it inhibits the development, the function, and the migration of autoaggressive Th17 cells. As you can see here, the Th17 cells are inhibited in their development, in their function, and also in their migration from spleen to the joints. We also studied the osteoclasts, and as you can see here, also the osteoclasts were inhibited in their development. We studied an in vitro model of osteoclasts, and as you can see, the decoction was going to inhibit the development of TRAP-positive cells, which are osteoclasts, and also the development of markers of osteoclasts, such as ACP5, CALS-R, MMP9, and CTSK. And also another feature of osteoclasts function, which is the phosphorylation of SRC. Also, this was the action of the decoction in vitro on osteoclasts, but also in vivo, all the markers of osteoclast generation were shut down, as you can see here, ACP5, MMP9, CTSK, and calreticulin. Okay, we made also a fingerprint. And as you can see here, we have many compounds in this decoction. Mostly these are auronol glucosides. And the most abundant is mesopsin 4-beta glucoside, T82, which also have the same features of the entire decoction, but it was less stronger. This means that it is able to inhibit the development of Th17 cells, but in a minor way compared to the entire decoction. So the decoction is better of the single main component of the decoction. This is true for the development of Th17 cells, but also, as you can see here, for the development of, for the inhibition of osteoclast generation. T82 is effective, but not so much as the entire decoction. Okay, so we can say that collagen in our mouse model can induce the development of arthritis because it induced the development and function of Th17 cells from spleen and the osteoclast from macrophages. And this brings to arthritis. But if you give the decoction of Artocarpus tonkinensis, you completely inhibit the development of Th17 cells and osteoclasts, and the arthritis reverses back, and the joints are again healthy. Sorry for this. So the take-home message for this first part is that Artocarpus tonkinensis decoction has a mechanism similar to that of very innovative biologic drugs, such as, for example, monoclonal antibodies against interleukin-17. And this decoction may be useful for other diseases that have the similar mechanism, such as arthritis, for example, multiple sclerosis, psoriasis, and the asthma resistant to glucocorticoids. Now let's go back to Delhi, and from Delhi let's go to Italy, to Rome first, but then we go to the South Italy, to the Sicily. Sicily is an island, very beautiful, and the main city in Sicily is Palermo. Palermo is an ancient and a modern city at the same time, very beautiful, one of the most beautiful cities in Italy, as you can see here from many monuments. And also is an ancient city because they have ancient religious traditions, such as also, you know, similar to India in some way. Here you can see people that for a religious ceremony, they undergo a complete trance. It's something that can happen only in that place. In this case, for the celebration of Easter. But Palermo has also a very prosperous vegetation heritage, and a lot of morace trees are present there. For example, a ficus here on the left, you can see a symbol of Palermo, which is an ancient ficus, and in the middle you can see another type of ficus, another species of ficus, is Ficus rubiginosa, that you can see here. Also, Ficus rubiginosa belongs to the Moraceae family, and we thought that maybe, as for Artocarpus tonkinensis in Vietnam, another member of the Moraceae family can in some way influence the immune system of our organism. So we made an extract from the leaves of Ficus rubiginosa and tested them against to see if they influence in some way the life of T lymphocyte, of CD4 T lymphocytes. Now, CD4 T lymphocytes derive from thymocytes. They go in the spleen or in lymph nodes and under a basic type of lymphocyte, which is Th0, and there they differentiate to different subtypes, Th1, Th2, Th17, T regulatory cells. We can do this also in the lab by stimulating the activation of Th0 cells through antibodies against CD3 and against CD28. And then we can drive the differentiation of Th0 towards the different subtypes, for example, Th1, Th2, Th17, and T regulatory cells. This is done by different cytokine cocktails, for example, interleukin 12 plus interleukin 4 to get Th1, interleukin 23 plus P40 to get Th2, and TGF-beta and plus interleukin 6 to get a balance between Th17 and T regulatory cells. So when you, in this experimental setting, you block the differentiation towards Th1 and Th2, you get only a balance between Th17 and T regulatory cells. Th17 is characterized by a transcription factor which is called ROR-C, and Treg cells are characterized by another transcription factor, FOXP3. Now, we made an extract, a methanol extract from Ficus rubiginosa, and we treated cells to differentiate towards Th17 and T regulatory cells. And as you can see here, as I said before, with a mixture of TGF-beta plus interleukin 6. As you can see here, all the methanol extract of Ficus rubiginosa was able to inhibit the differentiation of T regulatory cells, but not the differentiation of Th17 cells. So the methanol extract of Ficus rubiginosa moved the balance between these two kinds of cells, inhibiting T regulatory cells, but highlighting Th17 cells. The first conclusion is just this: Ficus rubiginosa methanol extract inhibits the differentiation of T regulatory cells, but not of Th17 cells. Then what we did, we made an additional fractionation of the methanol extract, and we got the hexane extract and the ethanol extract. And we tested this new extract in the same experimental setting as before. As you can see here, we analyzed T regulatory cells by means of flow cytometry. These cells are defined as CD4 positive, CD25 positive, and FOXP3 positive cells. And as you can see here, again, methanol extract was inhibiting, as we have seen before, and also the hexane extract was inhibiting the development of T regulatory cells. So the important compound for this inhibition are all in the hexane extract, but not in the ethanol extract. Now let me talk a little bit about the importance of interleukin-6 in the balance between Th17 and T regulatory cells. If you add interleukin-6, you favor the development of Th17 cells. But if you take off, withdraw interleukin-6, in this case, T regulatory cells prevail on Th17 cells. This balance is very important in our organism because Th17 cells promote autoimmune aggression, as we have seen in the arthritis, whereas T regulatory cells protect from autoimmune aggression, but T regulatory cells protect also from cancer. So if you want to treat autoimmune disease, you have to shut down, you have to upregulate the number of T regulatory cells, but if you want to protect from cancer, you have to shut down these cells. So what we did is to do the same experiment as before by subtracting, eliminating, withdrawing interleukin-6. And as you can see here, when you take off interleukin-6, you take off also completely the inhibition of T regulatory cells. So Ficus rubiginosa can inhibit still T regulatory cells, but only in presence of interleukin-6. The inhibitory effect of Ficus rubiginosa on development of T regulatory cells is dependent on the presence of interleukin-6. Of course, we tried also to see why there was this connection between Ficus rubiginosa and interleukin-6 by doing a gene expression profile. A lot of genes are modulated by this methanol and hexane extract. Here you can see some of them, for example, LIF, EFT, EFIT, TRIM30D, CD40, CD9, CCL9, sorry. But until now we did not identify the genes that were responsible for this interaction between Ficus rubiginosa and the interleukin-6. So before I leave all of you, I would like to thank my Vietnamese Italian friends that worked on this. For example, Professor Trinity Thuy, she's a professor of chemist in the Vietnamese Academy of Science and Technology, together with Bati Cham, Dr. Francesco Paolocci at the National Research Council in Italy. And as you see, there is also an intruder among them. Thank for your attention, but before I leave all of you, I would like to show you a small movie, because as someone already said, Pulok said before, in September there will be in Italy the conference, the 25th conference of the International Society of Ethnopharmacology, which I am the current president. And this conference will be in Italy, in Foligno, So all of you are invited, of course. We will have hopefully many people attending from India. And I would like to show a short video about this conference.
Foligno, the beating heart of Umbria and the navel of Italy, is a city with ancient Umbrian and Roman origins, now a vibrant center of commerce and culture. Here, St. Francis of Assisi embarked on his spiritual journey, and in 1472, the first edition of the Divine Comedy was printed. The city stands out for its majestic, noble palaces, witnesses to a glorious past. Palazzo Trinci, a treasure chest of Renaissance frescoes. Palazzo Orfini, the cradle of printing. Palazzo Candioti, the heart of the Quintana. And the Palazzo Comunale, a symbol of civic power. Foligno's spiritual soul is reflected in its churches and oratories. The Cathedral of San Feliciano, the Oratory of the Crucifix, the Church of Santa Maria Infraportas, and the enchanting Oratory of the Nunziatella, which houses frescoes by Perugino. Twice a year, Foligno transforms into the stage of the Giostra della Quintana, an event that revives Baroque splendor and engages the entire city in an exciting equestrian competition. The cultural scene is enriched by contemporary art, archaeological, and diocesan museums, celebrating the city's rich artistic heritage. Foligno is also synonymous with good living, featuring events like I Primi d'Italia and a lively, dynamic atmosphere along its bustling streets, full of shops and meeting places. The surroundings of Foligno offer breathtaking landscapes: Rasilia, with its natural springs, the Minotre Valley, with waterfalls and streams, the Abbey of Sassovivo, a place of spirituality, the Hermitage of Pale, immersed in nature, and the Colfiorito Park. Foligno also hosts 2 degree programs from the Faculty of Medicine and Surgery of the University of Perugia, one of the oldest universities in Italy and the world, physiotherapy and nursing. In September 2026, Foligno will welcome the 25th International Congress of the International Society for Ethnopharmacology, titled Ethnopharmacology and Transcultural Medicine. The event, held in the stunning venues of the San Domenico Auditorium and Palazzo Trinci, will focus on the interdisciplinarity and interculturality of ethnopharmacology, a scientific discipline at the crossroads of various fields of knowledge, based on the understanding and respect of traditional cultures. We look forward to welcoming you to Foligno.
Thank you. Let me end just thanking Nishit, which is helping me in these days, and also the driver that brought me last night from Delhi airport to here. And I would like to thank all of you. Thanks.
Thank you, Professor Delfino. We are now open to have some questions. Mike, please.
Good morning. Myself, Dr. Momita Kanjilal. I am from Clinical Research Division. Here I am, you can see. Yeah, it's here.
Okay.
Yeah. Sir, I'd like to ask you one question. Before that, we are very thankful that you have come over here and delivered a nice talk. Sir, in the field of arthritis, what you have presented, it is all the inflammatory arthritis which is shown. Sir, do you have any experience in non-inflammatory arthritis where CPPD crystals are involved, where we can see we can have something for those patients, because for inflammatory arthritis, many treatments are available, but for non-inflammatory arthritis, very less, few treatments are available. I am talking about calcium pyrophosphate phosphate crystals depositions in the joints, which most old age people are involved in that, geriatric cases.
Thank you very much for your question. It's a good question, but unfortunately, we don't have much experience with non-inflammatory arthritis. What I can say is that most probably the decoction of Artocarpus tonkinensis, it works only on inflammatory arthritis because of its action mechanism. As you have seen, the decoction inhibits Th17 cells, which is a feature of inflammatory arthritis. So probably the decoction does not work if there is no inflammation. And maybe you can try the other one, the Ficus rubiginosa, but I don't have any proof about this.
Any other questions from the audience? Please.
Hello, sir. Good morning. My name is Dr. Ram. I am heading the new product development in Patanjali for food and nutraceutical division. So this is a wonderful presentation and most welcome in India. So we have couple of products for this arthritis and anti-inflammatory. So did you check some couple of molecules as well, because this is like 100% herbal concoction? Did you check with like glucosamine or fonditin sulfate or methyl sulfonyl methane, which is very, very safe and treated as in like a nutraceutical and already proven in worldwide? So is any comparison with that as well?
Unfortunately not. I just studied these. It took to me 20 years actually to
All right. Okay.
because you know, with my Vietnamese friend, we started working on this in 2006, and we did some study on leukemia and on arthritis, but it's quite complex because probably you know that the collagen-induced arthritis is a very painful model.
Correct.
I mean, the mice must be very clean, because if they have, for example, a parasite in the intestine, they don't develop the arthritis. So it's a very painful model, and we just checked this compound, not other, but you know.
And this is the collagen, like type 3 collagen, denatured collagen, or how it is?
Sorry?
This collagen, whatever the collagen you have to type 1?
Is type 1 collagen.
Okay.
And you have to, usually any kind of mice can have this kind of disease, but the best one are DBA, DBA1 mice.
Okay.
They develop the arthritis after the challenge with collagen in about 50% of mice. And the other strain of mice, they get lower percentage of disease development. So if you have to use this, I strongly suggest that mice must be very clean and DBA1.
Thank you very much, sir. Thank you very much.
You're welcome.
Good morning, Professor.
Hello.
Sandeep Sinha, nice to see you again. So I just wanted to ask about the first plant, the Vietnamese plant. You said it basically acts very similar to the Th17 monoclonal antibody. Could you please elaborate more on that?
As I said, let me. No.
I can see you, Professor. Yeah.
Yeah, but I want to see you too. So the first row is, So yeah, as I said during the talk, the mechanism of action is really interesting because it's the mechanism of action of the biological drugs, such as monoclonal, anti-interleukin 17 monoclonal antibody. But this kind of drug, you know, the decoction is better because if you take a monoclonal antibody, you just target one small part of Th17 cell, the production of interleukin 17, which is important, but it's a limited target. But if you use, for example, a plant like this, you completely eliminate all the cell. So not only interleukin 17, but interleukin 22, interleukin 23. You know, the action is complete. While the action of a monoclonal antibody, which is important, is very good and, you know, put forward the medicine, the field of medicine a lot. But it's not enough because just blocking one target is good but not enough and favor also a lot of side effects. While if you eliminate all the cell, you decrease also the side effect and increase the efficacy. This is what I think. And thank you for, you know, welcome me again here in Patanjali.
Thank you, sir.
Thank you, Professor Delfino, sir.
Thank you so much, sir.
Please accompany me in thanking Professor Delfino with a loud clap.
Thank you.
Thank you.
Thank you very much.
I would now like to invite Professor Luis Felix Nothiez to present a keynote lecture on the topic From Molecular Evidence to Knowledge, AI-Driven Metabolomics and Literature Mining for Unlocking the Chemistry and Medicinal Plant. Professor Nothiez is a CNRS Junior Group Leader and Professor Chair based at the University Côte d'Azur and the International Institute for Artificial Intelligence, France. Professor Nothiez is a founding member of Holobiomics Lab, which involved in the development of experimental computational methods for studying the host microbial interactions. May we now invite Professor Nothiez to kindly initiate the keynote lecture. Let's welcome with huge round of applause.
Okay, 1, 2, 1, 2. Perfect. Thank you. Dear hosts, dear organizers, dear colleagues, dear audience, it's great to be with you today. I'm really happy to discover the research and share ideas. Thank you so much. So I'm leading a research group at the University Côte d'Azur that is composed of both chemists and software engineers. And you will see that in my presentation, that we are bridging chemistry and AI to develop new approaches. So there is an issue with the font. I'm sorry for the mess, but I will guide you into the process. Usually I never start with this slide, which is showing my track record. And as you can see, it's a long way from studying at the university to the PhD degree, the postdoctoral research. It's abroad. It's a very long process. And I wanted here to bring up how it started at the scale of my life. So in 2004, I traveled to India for 3 months when I was 18 years old. And I practiced Kalalipayattu hosted by a family of Ayurveda practitioners, and I lived with them for several months, and it was one of the best time of my life. And for me, it's very special to be here today. And at the time, I was practicing, so I was doing Panchakarma for the first stage, then I was practicing every day at 6 a.m. And after a month, I got injured badly, and I couldn't move. And so the grandfather offered me to come to the Ayurveda center to get some care. And he prepared the plaster with the plants, and I was discovering Ayurveda. It was very inspiring. And then I got this plaster. I could see the whole process. And then when I returned to my room, I couldn't move. And there was no internet, there was no cell phone. And the only books that were available were books on Ayurveda in English. And so I started to read for about a week, and it was really inspiring for me to discover this ancient wisdom. And because I have a scientific background from my high school, I was curious about, okay, what are the mechanisms of action? There must be very interesting discoveries to be made out of this. So this is myself. I don't have many pictures of the time, but this is myself, a young man, very excited about science, about the world, and this gave me a lot of. This shifted my perspective about cultural bias and openness on ancient wisdom. And so now back to my scientific career. The research question I'm pushing for is for the concept of the holobiont. So holobiont sounds like holistic. It's the same root, same Latin root. So it's the meaning that it's a continuum of life from the system biology standpoint. And so when we start from the genome, we can get all the way to the phenotype. And one major finding of the last decade is that every single living organism is associated with microorganisms that are part of its functionalities and that can really regulate how it operates. And so the major level for this interaction between the host, the macroscopic host, and the microbiome is the metabolome, small molecules that are like communication messages for it. And so, as I said, all life forms, including humans, are considered as holobiont now. And so if we want to study this organism, we need to combine many different omics approaches all together to have some representation of this living system. And if we add to this the interaction with other species, with the ecosystem, as well as the scale of environment and the scale of time also, with transgenerational effect and evolution. This is really fascinating. And so my specialty is metabolomics. So we don't see the pointer? Okay, let me do this. Up. Laser pointer, up. Yeah. Metabolomics, which is the science, the holistic science of studying metabolites in living systems. So I will discuss more on the progress made for metabolomics. This technique starts from a sample. It can be anything, it can be a plant sample, and we use an instrument, a technique called mass spectrometry that allows to ionize molecule so we can measure their spectra and get some structural information about their identity. And as you can see, typically we process the data and we start to do some statistical analysis to make sense of it. However, these fingerprints that we are collecting at scale, so it's really fast instrumentation that can collect thousands of spectra in tens of minutes. For many, many years, we were stuck because we had only few reference spectra, about less than 5%. However, the global community contributed by releasing additional reference spectra, enough so we could train deep learning models to improve the accuracy of what we can predict. And so nowadays, these advanced approaches are allowing us to annotate most of the spectra that we can collect. And of course, to make sense of the biological system, if we are restricted to just 5%, it's hard. But with more information, we can start to better propose hypotheses on these systems. So myself, with many colleagues from the US, from Germany, from Europe, we contributed to develop some of the software that are used nowadays to process the data and also to annotate them. So it's a long process to get all this paper together. It collaborative international effort. And essentially, we get from the data collected on the instrument down to some information that we can use on the biological system. Okay, this is more problem. Oh, it seems like most of my slides are, we will see. Okay, I will explain. Yes, it's a new language. So this is the pipeline essentially. So starting from the plant, we extract, and you are familiar with this at Patanjali Research. We extract the plant, we perform some mass spectrometry analysis by liquid chromatography. So we separate the constituents and we collect the fragmentation spectra on them. And as I have explained, we can deduce the molecular information, molecular formula, the structure, and nowadays, if we don't have the structure, we can have the chemical classes for most of the signals. However, we have these efficient approaches, but it's very complex to interconnect the information. So we have like heterogeneous set of information that we have to aggregate efficiently. And so this is a major challenge. And so how do we make sense of this